Assalamu alaikum everyone. This is me, Dr. Faiza here, with the second session on special senses. The topic I have here today for discussion is receptor and neural function of retina. In the previous session, we had seen the basic principles of the optics of vision, and today we are discussing the receptor and neural function of the retina. For the session, I will be using references mostly from the textbook of Physiology, Guyton and Hall. It's chapter number 51. And some references from Genome Physiology, chapter 10, Sherwood Human Physiology, and BRS Physiology also. So let's begin our discussion about the retina. To move up towards its physiological functions, first we have to look at the relevant anatomy of retina. In front of you is the anatomy of retina showing different layers of retina. Starting at one end, if we start at this end, we have number one, pigmented layer of retina. Pigmented layer of retina is having melanin inside its cells, because of which it is known as pigmented. Also, it is a site of storage of vitamin A, an important precursor of photopigments which are necessarily required for phototransduction. So this layer acts as a storage form of vitamin A2. Because of melanin and pigmentation present within this layer, it prevents scattering of the light. It absorbs the light. Otherwise, once the light strikes the retina, it would have scattered all around and the visual field would have been simply bleached. Because of this pigmented layer of retina, the light is absorbed and it makes a meaningful image on the retina. For example, among albinos who have reduced or absent pigmentation of melanin in their retina, they have very reduced visual acuity in the bright light. Moving on, the second layer is composed of photoreceptors, the actual receptors which are receiving the light to make the image on the retina. These are of two types, rods and cones. Rods and cones have photopigments in their outer segment, and their nucleus is present in their inner segment. There is the third layer, outer limiting membrane, which is separating the outer segment of photoreceptors from their inner segments. This is outer limiting membrane. The nuclei of photoreceptors, they are forming fourth layer, outer nuclear layer. These photoreceptors, once they receive light, they hyperpolarize, they send their message to the next layer of cells, bipolar cells. In between the bipolar cells and photoreceptors, you can see these horizontal cells. These are forming outer plexiform layer, the horizontal cells. Bipolar cells, they are forming the inner nuclear layer. Inner nuclear layer bipolar cells then further send the message toward the ganglion cells. Again, you can see in between the bipolar cells and ganglion cells, there is an other layer of cells which are having horizontal communication with both the bipolar and ganglion cells. These are the amacrine cells. They are forming the next layer, inner plexiform layer. Ganglion cells are forming the layer ganglion cell layer. The axons of ganglion cells are actually forming the optic nerve, the next layer, stratum optica. And lastly, we can see the inner limiting membrane. Inner limiting membrane is composed of the glia cells, molar cells, which are responsible for holding the neural elements of retina together in the place. This is the limiting membrane which separates the neural retina from the vitreous humor. So if we look inside the eye, in front, if this is the lens and behind is retina, light goes in this direction. It goes through all these layers reaching the photoreceptors, and lastly the pigmented layer. So first of all, light falls this, it goes through this direction. But the transmission of nerve signal, it is in the opposite direction. It goes from photoreceptors to bipolar to ganglion cells to the optic nerve. So direction of light and direction of phototransduction is opposite to each other. The layer of retina facing the vitreous humor is this one, inner limiting membrane. Horizontal cells and amacrine cells both are responsible for the lateral inhibition, as we have seen in the sensory physiology, motor physiology. To sharpen a signal, the neighboring areas are inhibited simply by lateral inhibition. In the outer and inner plexiform layer, cells responsible for this lateral inhibition are horizontal cells and the amacrine cells. The blood supply of retina, you will be reading more in detail in the anatomy. For the functional point of view, the retina neural layers are mostly supplied by the central retinal artery till the photoreceptor layer. Photoreceptors, specifically their outer compartments, this and the pigmented layer, these are mostly supplied by the choroidal blood vessels rather than by the central retinal artery. In case if there is retinal detachment, what is retinal detachment? Retinal detachment is the separation of pigmented layer of retina from the neural layer of retina. So here the retinal detachment occurs. If retinal detachment occurs, the retinal artery will keep on supplying this neural tissue. Choroidal vessels will keep on supplying the pigmented layer. Most vulnerable to destruction and ischemia will be the photoreceptor layer. And if retinal detachment is not treated well in time, the photoreceptors will be mostly damaged because of the decreased blood supply. That was the layers of retina. Like among the cardiac cells and smooth muscle cells, the neural components of retina are connected to each other with the help of gap junctions. A very important thing to know over here, because this is what is going to be linked to their electric potential later on. All these are neural elements are connected to each other layer by layer with the help of gap junctions. Fovea centralis, a point in the retina where the visual acuity is the highest. This is responsible for acute and detailed vision. Where we can find this fovea centralis? We can find it in the center of retina, almost 1 millimeter square surface area. This is fovea centralis. If we look at its histological characteristics, we will find only the cones inside the fovea centralis which are responsible for detailed color vision. There are no rods present in the fovea centralis. Secondly, the many layers present in front of the photoreceptors, they impede the entry of light to the photoreceptors. They slow it down, they impede it. But in the fovea centralis, as you can see in this image, rest of the layers have moved to the sides. Light is almost impinging directly upon the photoreceptor layers, upon the cones inside the fovea centralis, which also helps in increasing the visual acuity in this layer. Maximum visual acuity is found within the fovea centralis, but this is having very low sensitivity to the dim light. It will respond very well to the bright illumination, but sensitivity is less. Sensitivity increases as we move towards the periphery, because towards the periphery, the number of rods increase and number of cones decrease. So one thing we can decide over here, rods are having more sensitivity and cones have more acuity. We will be discussing in detail in the coming slides. Rods are more present in the periphery of the retina, where cones are majorly more abundantly present within the fovea centralis. Another characteristic of fovea centralis that we can discuss over here, that cones in the fovea centralis have one-to-one communication with the bipolar cells. There is almost equal number of cones and equal number of bipolar and ganglion cells in the fovea centralis. Whereas in the periphery of the retina and specifically for the rods, many rods, they converge on a single bipolar cell which send the message to the ganglion cells. That increases the sensitivity of rods but decreases the acuity. The one-on-one communication of cones with the bipolar cells at the level of fovea increases its acuity at the expense of its sensitivity. Another special component at retina, optic disc. It's completely opposite to that of the fovea centralis. Fovea centralis had the maximum visual acuity, a smaller location in the center of retina, whereas optic disc is the point in the retina where there are absolutely no photoreceptors. There is no vision at this spot. This is known as blind spot, physiological blind spot. This is completely normal. We all have this physiological blind spot. Why is this created? Because this is the point in the retina where the optic nerve leaves the retina. We find it almost 3 millimeters medial and slightly above the posterior pole of the eye. If in the posterior pole center portion is fovea centralis, just medial and little above to it, you will find the blind spot, the optic disc, the physiological blind spot. You can see in this image shown over here, this is the optic disc and this is fovea centralis. Macula lutea is also linked with the fovea centralis, and the maximum visual acuity is in the portion of fovea centralis, which is also a component of macula lutea. We have this handheld device in our clinics known as ophthalmoscope, which can be used to visualize the fundus of the eye, means the interior surface of the eye, including the retina, optic disc, macula, fovea, and posterior pole, the arteries, veins, and superficial layers of the retina. The image you can see, we can see all this with the help of this ophthalmoscope at the bedside. This can help us diagnose many diseases. For example, in glaucoma that we discussed in the previous session, the normal optic disc shown over here will be converted to this one in glaucoma. What you can see, the optic disc has become pale. The blood vessels you can see have become tortuous. Their support has decreased. There is cupping of the optic disc. Normal cup to optic disc ratio is 0.5. If it is increases, one of the reasons is glaucoma. Now, what is cup to disc ratio? The whole circle, this is fovea centralis, and in between this is a smaller portion. The ratio of this circle to this should be normally 0.5. If the ratio increases, that means the cupping of the optic disc has occurred. Moving on towards the details of the photoreceptors. We will be looking into the further details of other layers too, but mostly we are going to focus upon the rods and cones in the sessions, which are the photoreceptors for receiving the light signal. In front of you, there is rod and cone, evident by their shape. This is cone shape and this is rod shape simply. The diameter of cone is smaller, 1.5 micrometer at the fovea. Towards the periphery, the diameter of the cone starts increasing. Rods are, however, rod-shaped and of almost consistent diameter throughout the retina. If we look at their subcomponents, both rods and cones have an outer segment. In the outer segments, they have shells in their membranes, sacs or discs in the cones and rods. They are there to have photopigment. photopigment is studied in the form of transmembrane proteins in the shells of rods and cones within the outer segment. Inner segment of rods and cones have nuclei. Number 2, abundant mitochondria. Nuclei and abundant mitochondria. Lastly, there is a synaptic terminal which will synapse with the bipolar cells. This slide is showing the photopigment, rhodopsin or color pigment. Rods have the photopigment rhodopsin, whereas cones have the photopigment scotopsin. We will be looking into the details of rhodopsin and scotopsin in a while. The rods help us look at the things even in the dim illumination, even in the dark. The rods are mostly responsible for the black and white vision, known as scotopic vision, whereas cones respond to the bright light. They have high visual acuity. They help us discern the details of the subject. They help us discern the color composition of the objects. So they are mostly related to the photopic vision. The presence of both rods and cones in the retina associated with the scotopic and photopic vision. This is known as duplicity theory. In this diagram from Genome, it's showing the percentage distribution of cones and rods in the retina. Here you can see this is Fovea Centralis, the center of retina. Here this blue line is showing the abundance of cones and red one is showing the abundance of the rods. At Fovea Centralis, you can see cones are most abundant and their abundance is decreasing as we go towards the periphery on either side of the retina. Whereas rods are equal to 0 at the point of Fovea Centralis and their abundance increases on either side of the retina. Now that we know the photoreceptors, rods and cones have the photosensitive pigment in their outer compartment. What that photosensitive pigment is, that is rhodopsin in rods. You can remember it by the R, and photopsin in the cones. These pigments are further composed of two components. One, 11-cis retinal, and secondly, scotopsin. Rhodopsin is further composed of these two things. 11-cis retinal is an isoform of vitamin A. Vitamin A in the pigment layer is stored in the 11-trans retinol form, which can be converted into 11-cis retinal with the help of isomerases. So rhodopsin has essentially the presence of isoform of vitamin A and also the photopsin. If vitamin A is absent, the photopigments will be decreased and that can lead to night blindness. Let's move on towards the retinal rhodopsin visual cycle in front of you. What happens in the dark state? The photopigment is present in the form of rhodopsin. Right now we are looking at this visual cycle in the rods. In the dark, the photopigment is found in the form of rhodopsin. As the light falls on the rods, this rhodopsin is decomposed into sequential substances in front of you. Bathorhodopsin, in a while into lumirhodopsin, then metarhodopsin 1 and then metarhodopsin 2. So in a few seconds you will see the rhodopsin is decomposed into metarhodopsin 2. This metarhodopsin 2, this is the active form and it quickly decomposes. This activated rhodopsin decomposes it to scotopsin and 11-trans retinol. Now, next the signal will be transmitted. We will be reviewing in the next cycle. In the next slide, This 11 transretinal, if it is excessive, it might go stored in the pigment layer, and if it is less, the more vitamin A might be converted into transretinal and then cisretinal. Ultimately, when the light energy is removed, this 11 cisretinal will combine again with the scotopsin to reform the rhodopsin. So this cycle keeps on continually being happening inside the rods. Rhodopsin being formed, rhodopsin being decomposed. More the dark, more rhodopsin will be present. More the light, more rhodopsin will be decomposed. Vitamin A deficiency, as we discussed in the previous slide, can lead to night blindness. This is mostly caused because of the nutritional deficiency that builds up over the years. In front of you is the source of vitamin A in the dairy products, and beta carotene, which is a precursor of vitamin A, can be obtained from the green leafy vegetables or yellow orange fruits and vegetables. For example, carrots are very famous for providing vitamin A. In the absence of vitamin A, when the rhodopsin photosensitive pigment will decrease, first of all, it may lead to night blindness, which is also known as nyctalopia. It can also lead to dry cornea, known as xerophthalmia, dry retina. It first alters the rod function. This is why it first leads to night blindness, because rods are responsible for the dim light vision, black and white vision. Later on, if continued, it may lead to cone degeneration followed by degeneration of other neural layers of the retina. And if diagnosed and treated, single treatment dose may reverse the symptoms. But the condition is that treatment is initiated well in time before the permanent damage is caused. Moving on towards the phototransduction, back to this. This is the ionic channels in the inner and outer segment of the photoreceptors. What happens usually in the state of dark, there is a sodium potassium ATPase, which is present in almost every cell of the body. It keeps on moving sodium out of the rods and potassium into the rods. It is an electrogenic pump. It creates electronegativity inside the cells because 3 sodium ions are moved out in comparison to potassium ions are moved inside. Number 2, what happens inside these receptors, that potassium is continuously sent out of this segment, and these channels are moving, these cyclic GMP gated channels are moving sodium into the outer segment of rods and cones. This is the magnified version of this in state of dark. These cyclic GMP gated channels are open and sodium is continuously going back into the cells. This re-entry of sodium into the cells is helping keep the potential of these receptors little bit depolarized. Normally we have minus 70 millivolts, but rods and cones in the dark without the light stimulus, because of the entry of the sodium, they are staying in this partial depolarization state, minus 40 millivolts. What happens when the light comes? These sodium channels are closed. Sodium can no more enter into it. Now potassium ions are leaving through these channels. Sodium ions are leaving through these channels, leading to increased negativity inside the cells, and the polarity of the cells becomes minus 70 to minus 80 millivolts. Hyperpolarization occurs. This is one of the very few examples in the body where stimulus instead of depolarization is causing the hyperpolarization, and reason is in front of you, the closure of CGMP gated sodium channels. Now let's have a look why these channels are closed in state of light stimulus. When the light comes, the rhodopsin is decomposed. It is decomposed, metarhodopsin is formed, and that leads to stimulation of another G protein known as transducin. This transducin protein stimulates the phosphodiesterase, which decomposes the cyclic GMP to inactive 5-GMP, resulting in the closure of the sodium gated channels. So simply when the light falls, the CGMP level is decreased. That leads to the closure of sodium channels. That leads to the hyperpolarization of rods and cones. Duration of the receptor potential produced here will be proportional to the stimulus intensity. The more the light is intense, the more the sodium channels will be closed. Once there is light, CGMP is reduced, sodium channels are closed, there is hyperpolarization, and the message will be sent to the bipolar cells. Now, how will this activity be reverted back to normal? There are two postulated mechanisms for this. One is rhodopsin kinase, which will deactivate this transducin. Number two is there is a protein beta-arrestin present in the cytoplasm that will combine with this transducin, making it inactive, and the closure of sodium channels will be reverted. Involvement of G protein anywhere in the body that amplifies the signal. So just a small stimulus, even the dim light when falls on the rods, increases the amplified signals with the help of the G proteins, helping us to see even in the starlight. We can see in the sunlight, but still we can accommodate to see in the starlight because of this amplification mechanism present within the rods. So if we summarize the phototransduction across the rods, whenever there is light falling on the rods, there will be structural change in the photopigment. The structural change will lead to formation of activated rhodopsin, which will cause activation of transducin and phosphodiesterase that will decrease the cyclic JMP. Sodium channels will be closed. There will be hyperpolarization, and hyperpolarization will do what? Hyperpolarization at the synaptic terminal of rods will decrease the entry of calcium. Calcium channels will be blocked. Once the calcium channels are blocked because of hyperpolarization, release of glutamate, which was the excitatory neurotransmitter being released from the synaptic terminals, will be inhibited. So something exceptional I would like to emphasize again. Hyperpolarization is caused instead of increase in the neurotransmitter release, that neurotransmitter release is decreased. Decreased release of glutamate, and that will be then conducted to the bipolar cells. And the message will be carried forward in the rest of the layers of the retina. So if you are asked about the phototransduction through the rods, you can give this detail. If you are asked about the retinal rhodopsin visual cycle, you can give this detail. If you are asked about the functioning of ionic bases of rods and cones, you can give this detail along with this detail. So moving on, phototransduction of the cones. Cones again have the photopigment called photopsin, including again the aldehyde portion retinal, and the other portion instead of scotopsin here is the color sensitive pigment, red, green, blue, for the three cones. There are three types of cones: red cones, green cones, and blue cones, which are having their sensitivities to different wavelengths of light, which is in front of you. Blue light to the short wavelength, green to intermediate, and red have their absorption at the large wavelength of light. Rods are having intermediately at 505 nanometer in between the blue and the green cones. We have now seen the phototransduction across these photoreceptors. Let's talk about the light and dark adaptation. Very important mechanism. If we go out from our room directly into the sunlight, for a while we cannot see clearly, but within seconds our eyes adapt to that bright light, and now we can see very easily and comfortably in the sunlight. How this happens? The reason is, once we are exposed to bright sunlight for a while, most of the rhodopsin is already converted into scotopsin and 11 transretinol. Once the less rhodopsin is available to decompose to respond, the light will not be bothering us anymore. That will decrease the sensitivity to light because already most of the rhodopsin is decomposed. And that decomposed rhodopsin will be evident in the form of increased storage of all transretinol in the pigment layer. In comparison, dark adaptation. If you are in the dark for a while, most of the scotopsin and cis-retinol will have been combined to form most of the rhodopsin. There will be decreased vitamin A, decreased all transretinol in the pigment layer. The maximum possible will have already been converted into the rhodopsin, so sensitivity to light is increased. The mere amount of light present is enough to stimulate the rods here, helping us to see inside the dark. This is what happens once we are in the dark or in the light for a while. What happens if we go into the light from a dark area? For example, you are watching a movie in the cinema and now you go outside from the dim light to the bright sunlight. What will happen in the first few seconds and minutes? Your rhodopsin will be too much because you were there in the dark for 3 hours watching a movie. So your rhodopsin is not decreased. Initially, your field visual acuity will be decreased. But if you look at this graph, which is showing the time duration on the x-axis and retinal sensitivity on the y-axis, you can see just in 1 minute, the cones adapt and retinal sensitivity has already changed by 10 times. If you look at the sensitivity at 20 minutes, you can see it has almost 6,000 times changed already. And at the end of the 40 minutes, you can see it has changed more than 10,000 times. How does this happen? In the dark, in the light. For the dark adaptation, the sensitivity of retina, it increases. For the light adaptation, the sensitivity of retina, it will decrease. Same will happen, but in the reverse direction, which helps us to adapt both the cones and the rods. If you look at the graph again, cones are first to adapt. They are rapid. The rapidity of adaptation is much more than that of cones. But if you see the potency, the rods adapt much more as compared to the cones. So if you talk about the potency, rods are more potent in adaptation, but cones are more rapid in adaptation. In front of you is the dark adaptation curve. Just assume that you were enjoying in the bright sunlight playing cricket and now you have just entered inside your bedroom and there is no light, electricity is gone, but still in a while your eyes will start seeing things around you because of this adaptation. First cone adaptation and later on rod adaptation. How will they adapt? There are 3 ways of adaptation for dark and light adaptation. Let's have a review of all these conditions. One is the change in concentration of rhodopsin that we already discussed. For dark adaptation, more and more rhodopsin will be formed. This will increase the sensitivity of retina. In reverse, for light adaptation, more and more rhodopsin will be converted into transretinol. This is going to take several minutes. As we saw in the previous graph, the maximum adaptation is occurring at the 40 minutes. Although it takes much time, but it's the most potent mechanism. Number 2, change in the pupillary size. Yesterday we saw pupillary dilator and constrictor muscles supplied by the sympathetic and parasympathetic, which can change the pupil size. It can decrease up to 1.5 millimeter and increase up to 8 millimeter, which can change the depth perception. So for dark adaptation, the pupils will dilate. For light adaptation, to allow less amount of light to enter in the pupils will constrict. This is a rapid mechanism and occurs within the fraction of a second. Lastly, the neural adaptation. If there is dark, throughout the visual pathway, the sensitivity of nerves will increase. More and more facilitation of nervous transmission will be there, but to adapt for the light, the neural transmission throughout the visual pathway will be inhibited. There will be decreased signal transduction across the visual pathway till it reaches the visual cortex. This also happens very rapidly in fraction of a second, but it is much less potent, much less important as compared to rhodopsin mechanism. So for dark adaptation, light adaptation, there are three mechanisms, among which most important one is the amount of rhodopsin and amount of rhodopsin being decomposed. Cones are more rapid, rods are more potent. So for black and white vision, dim light visions, rods are there to help us more, but cones are there to help us until the rods have adapted to it. Moving on to the next topic, the color vision. We saw the duplicity theory, photopic and scotopic vision, among which we have seen the transduction across the rods. Now we are moving towards the color vision. There is a trichromatic theory. Young-Helmholtz theory of trichromatic theory of color vision, which says that human eye can detect almost any gradation of color just by mixing any of these three colors. The primary colors, red, green, or blue. If mixed in appropriate amount, human eyes can see any of the secondary colors. For the detection, we have three types of cones: blue cone, green cone, red cone. We have seen their wavelengths: small, medium, and large. Their chromosomes having gene for the pigments is X chromosome for green and red cones, whereas for blue cones it is present on chromosome number 7. For the rods it is present on the chromosome number 3. By appropriate combination of stimulation of these three cones, any color can be perceived by the human eye. Also, there are other factors which can impact this. For example, degree of illumination, hue intensity, saturation of the color, and the phenomenon of opponent color. What is that? If you are seeing red across white, there is a red flower in front of you and the room is illuminated by white light, you will see it as red flower. If you're seeing a red flower and the light source is also red, you might see it as pale pink. So the illumination is mattering. The opponent colors against the background and the illuminating colors, that also matter. For now, let's stay simple. If everything else is kept constant, how these colors are perceived? Let's have a look. For orange color in the spectrum, if you see, it is stimulating mostly the red cones, somewhat green cones, but not at all the blue cones. To perceive blue color, sorry, this is the typographical error, only the blue cones will be stimulated. There will be no stimulation of green or red cones. For yellow color, again, red and green cones are activated, but in different combination. For green, there is different combination. So by appropriate combination of stimulation of these three cones, any color will be perceived. And if all the three cones are collectively stimulated in equal proportions, white color can be perceived. Equal stimulation of all three cones. There is another interesting phenomena, black color. Black color is actually a color. It's not the absence of color, because blinds don't see the black color. Actually, they see nothing. If we see at the defects of the color vision, That is known as color blindness. Mostly this is X-linked. Red color blindness and green color blindness, whereas blue cones are not operated upon by the X chromosome rather than by the somatic chromosome, so that is not X-linked. X-linked and recessive. So it will X-linked, so it will be found more among the men. Color blindness can be of three types: monochromats, dichromats, trichromats. Trichromatic vision is actually the normal vision in which all the three cones are working fine. Dichromatic vision is in which two cones are working fine, just one cone system is defective. The cone system which is defective, that is the name of this disease. Protanopia, red cone is defective. Deuteranopia, green cone is defective. Tritanopia, blue cone is defective. You might have come across the phenomena, the terminology of red-green color blindness. So red-green color blindness again is a type of dichromatic color blindness in which the person is unable to differentiate red color from the green color. Tritanopia is the most rare form of the color blindness. Red-green color blindness is the most common form of it. Lastly, the monochromats are those who have only one functioning cone system and two of the other cones are defective. Color vision can be assessed by using these Ishihara charts. On the bedside simply, which use these color palettes and ask them to see what's written in these plates, the colorblind person will be seeing either no number or a different number from the normal healthy eye. Color vision loss or color blindness is not only caused by the defect at the level of cones, but it can also be caused by a defect at the level of visual cortex, specifically in the V8 portion of the associative visual cortex, and that disease is known as achromatopsia. We will be looking into the detail of the visual cortex in the tomorrow's session. So moving on towards the neural function of retina, we have seen the photoreceptors in detail. Now let's have a look at the successive steps in the retina for transduction of light energy to the brain. Once the photoreceptors are activated, they are hyperpolarized, there is decreased rate of release of glutamate from the photoreceptors. The message is next sent either directly to the bipolar cells, as you can see here, or it can be sent to the horizontal cells, as you can see here. The lateral plexus, that horizontal cell might communicate with the bipolar cells. So both of these ways can happen, and the horizontal cells are hereby sharpening the image formation by the phenomenon of lateral inhibition. Again, if I emphasize the difference of fovea from the peripheral retina, at the level of fovea, more and more direct connection will be found in the cones and the bipolar cells. Going towards the periphery, more and more indirect connections will be formed, more and more number of rods will be converging on a single bipolar cell. It can be one-to-one, it can be many-to-one communication. Again, showing the phenomenon of lateral inhibition by the horizontal cells. This photoreceptor is activating the bipolar cell and collaterals are activating these horizontal cells to inhibit the surrounding cells. The neurotransmitter at the level of rods and cones is glutamate. Amacrine cells are found to have many types of neurotransmitters. There are about 20 types of amacrine cells discovered till now. The neurotransmitters GABA, glycine, dopamine, acetylcholine, and dopamine. We are not supposed to remember all of these. Thing to remember is this. There was one exception that we discussed, that rods are hyperpolarized in response to the stimulus rather than being depolarized. The other difference here is all of the transduction of message starting from the rods and poles till the ganglion cells is with the help of electrotonic conduction, not in the form of action potential. As we all know, receptor potentials are graded. They may be increasing with the response to the increase in the intensity of the stimulus. They will lead to the production of action potential only when there is the threshold potential achieved. Whereas action potentials, they follow all unknown phenomena. Throughout the retina neural pathway, the action potentials are only generated in the ganglion cells when they have to send the message via optic nerve far away to the visual cortex. Before that, there is the conduction of receptor potential in between the neural layers of retina with the help of the gap junctions that we emphasized in the earlier slides. The gap junctions are present in between the neural layers because it is going to help in the conduction of the electrotonic potential. This is showing the phenomenon of lateral inhibition. This is excited area around which this is the inhibited area because of lateral inhibition. So if we look at the types of cells in the retina, rods and cones, they hyperpolarize in response to light. Horizontal cells, they are also inhibitory cells. They will be having inhibitory neurotransmitters to cause the lateral inhibition. Bipolar cells are of both types. They can be excitatory and they can be inhibitory. Amacrine cells, mostly they are causing lateral inhibition. These are also inhibitory. Ganglion cells have to form the action potential by all or none phenomena and send via the optic nerve to the visual cortex. If we look at the proportional number of rods, cones, and ganglion cells, on average, there are 60 rods converging on one ganglion cell in comparison to cones converging on each ganglion cell. Rods are so many as compared to the cones. Convergence is much more among rods as compared to the cones. Retinal ganglion cells are of two types among humans: parvocellular ganglion cells, magnocellular ganglion cells. The parvocellular ganglion cells, they will have smaller receptive field, means they will have less sensitivity but more acuity. In comparison, magnocellular ganglion cells will have larger receptive field. They will have more sensitivity and less acuity. Parvocellular ganglion cells will slowly conduct the impulses. Magnocellular ganglion cells will have faster conduction of impulses. Parvocellular cells will respond to sustained stimuli, whereas magnocellular response cells will respond to transient stimuli. More velocity of conduction, rapid change in the visual field stimuli. Parvocellular ganglion cells are majorly involved in the color vision with the cones, whereas magnocellular ganglion cells are majorly involved in the black and white vision. The color vision, cones, parvocellular ganglion cells have less sensitivity. The rods, the magnocellular ganglion cells have more sensitivity. Once the message is conducted to parvo or magnocellular ganglion cells, separate nerve fibers will be carrying the message to the parvocellular or the magnocellular layer of the lateral geniculate nucleus. So starting at the level of retina, the black and white and color vision is separated from the ganglion cells, from the photoreceptors, ganglion cells, the thalamus and cortex. The pathways will be kept differently. The color vision pathway will have low sensitivity, more acuity, slowly conducting. Magnocellular pathways will be faster conducting. They will detect the rapid changes in the stimuli. They can have very low threshold for their stimulation, very high sensitivity, and they can help us in the black and white vision. So emphasizing same things again in different forms at different levels of the retina. There is a third type of ganglion cells too, which do not have photopsin, scotopsin, rhodopsin. They have melanopsin. Where do they project? They do not go to the visual cortex. Rather, they project their ganglion cell neurons to the suprachiasmatic nucleus of hypothalamus, and they are there to regulate the circadian rhythm. Once the visual image is formed on the retina, the bipolar cells and ganglion cells, they respond in two ways. In front of you, there is one type of cells which have on center, and the other type of cells have off center. Secondly, the surrounding is opposite towards inside. Those who are on center have off surround, and those who had off center have on surround. What does it mean? It helps in keeping the visual acuity precise. Sharp contrast borders formation, one of the functions of lateral inhibition. Why does this happen? This explains that some of the bipolar cells and ganglion cells, upon stimulation by rods and cones, are stimulated and some are inhibited. Those who are stimulated, their peripheries will be inhibited. Those who are inhibited, their peripheries will be stimulated. Now, who will decide which bipolar cells are going to be stimulated and which bipolar cells are going to be inhibited? This is additional information. There are two theories that can explain this. One is the very simple one. It is postulated the bipolar cells directly stimulated by rods and cones will be stimulated. They are on center, off surround, whereas the bipolar cells stimulated indirectly by the horizontal cells are of course going to be inhibited. They are off center, on surround. A very simple explanation. The other more widely accepted explanation is that the glutamate being released at the synaptic ends of rods and cones had two types of receptors: ionotropic receptors and metabotropic receptors. Ionotropic receptors are which lead to opening of voltage gated channels upon binding of the glutamate, and metabotropic receptors are those related to the G proteins. The bipolar cells having ionotropic receptors are inhibited by glutamate, whereas the bipolar cells having metabotropic receptors are stimulated by glutamate. Different type of receptors, same neurotransmitter, different response will occur. Now what happens if there is dark? There is more and more glutamate formation. So these bipolar cells will stay inhibited. In comparison, these will stay stimulated. I'm sorry. When the light comes, glutamate production is decreased. Now these ionotropic receptors containing bipolar cells will be stimulated, and these will now be inhibited in the absence of glutamate, and their peripheries will be opposite. So we will call these bipolar cells on center off surround, on center off surround and off center on surround, and their image will be perceived like this in case of light. This is on center off surround. This is off center on surround. So because of difference in the receptors present on the bipolar cells, glutamate will have different effects on the bipolar cells and they will respond differently. This is summarizing the same thing, the whole of the phototransduction in the presence of dark and in presence of the light, what we have already discussed. In some bipolar ganglion cells, on center off surround, in other off center on surround, ultimately it will help in making a sharp contrast image. Also, the color signals are transmitted by the ganglion cell layer, and they also respond the same way on and off phenomena. Till now, we have seen many differences between the rods and cones starting from the session till now. Let us summarize all those. There are many more rods as compared to cones in the retina. Rods are more in the periphery as compared to cones, which are more abundant at fovea. Rods have more sensitivity. They are involved in dim light, night vision, but they have low acuity. In comparison, cones have high acuity. They are involved with day vision, but at the cost of low sensitivity. Rods converge much more as compared to cones in the retinal pathways. Ultimately, rods are involved in black and white vision and cones are involved in the color vision. So that was all for the phototransduction across rods and cones and other neural and receptor functions of retina. I hope it was useful for you all. If you have any questions, you are more than welcome to ask us. And till the next session, take a very good care of yourself. Allah Hafiz and goodbye.